Hey, everyone, and welcome to episode 218 of the Tick Bootcamp podcast. The title of today's interview is There's Hope, an interview with Dr. Timothy Haystead. My name is Richard Johannesson.
And I'm Matt Sabatello.
Matt, our podcast profiles the victories of individuals in the battle with Lyme disease, but I often find myself feeling hopeless about the prospect of a global solution to this growing pandemic. But today, after meeting Dr. Timothy Haystead, I feel differently.
Rich, Dr. Haystead was so humble, but in our opinion, he's a genius. He discovered this drug discovery platform that he used in the cancer world, the HIV world, the autoimmune world, and even for COVID that he's now brought to the tick-borne disease community. He's identified two major problems with chronic Lyme. The first is that there's no way to accurately diagnose a chronic infection. And the second thing is there's no way to properly treat chronic Lyme as antibiotics are not effective. So he's using this technology and bringing it into the tick-borne illness community to now develop proper testing and treatments for chronic Lyme disease.
Matt, I really appreciated the humility that this genius showed during the course of this podcast. And in particular, I was really moved when he said to you that he did not want to be considered a medical star because that is a really dangerous event. And what he said was that when we create these medical stars, in many cases, what happens is they stop us from having the diversity of research that we need. They essentially become gatekeepers. And that was something that allowed us to have a conversation with him about how for 30 or more years, the medical community has limited Lyme research because there were some stars very early on that placed restrictions on the research that so many people had done, such as Dr. Alan McDonald. So Matt, I'm really excited to introduce Hope and Timothy Haystead to the Tick Bootcamp podcast. Hey, Professor Haystead, and welcome to the Tick Bootcamp podcast.
Nice to be here. Nice to meet you.
First, Dr. Haystead, we've noted an accent, which is unique, at least to Long Islanders. Matt and I have our New York accent. So talk to us about where you're from.
You guys got the accent.
That's right.
Not me. I'm originally from the UK. I grew up in a town called Shrewsbury, where actually Darwin went to school. So that's probably why I'm inspired to work in biology and biochemistry. And then joined the rest of my education. Is that right? Or where I came from?
So talk to us. Talk to us about what inspired you to become a biochemist and what the path was to becoming a doctor of biochemistry.
Yeah, I mean, of all the things I took, chemistry, physics, math, that was the easy one. So I could get good grades. No, I was kind of interested in, just what life was about, and how it came to be. And then didn't really imagine you could make a career out of that back in the where were we? Back in the 70s. No one remembers the 70s.
I do.
It was too much of a good time. Well, yeah, that's not good then. You didn't have a good enough time. So, but anyway, so that was, it was an interesting career. It didn't seem like a job to me somehow, because you were kind of fussing around, playing with bugs and things and looking down microscopes. And then I got interested in the idea of drugs and how they're made. And so I took a degree in biochemistry and physiology at the time, and in Cardiff University, which is in Wales. I don't know if you know where Wales is, but it's if UK, it's consisted of a number of different nations, really, why it's called the United Kingdom. And I lived in Shrewsbury, which is on the Welsh border, and I went to University College Cardiff, which is in South Wales, but really enjoyed that. And then went on from there. You take so there's an interesting contrast between the US and the UK. Right? So you go to the UK and you take a degree, and you basically have fun for 3 years. And then at the end of the last 2 weeks of the 3 years, you take these exams, and everybody goes crazy because they cram they haven't been paying attention, and your whole life is dependent on what happens next. Some people jumping off buildings, they're like stressed. Whereas here, you do it gradually, and I think that's it. The system here is better. Real stressful there. Anyway, so then you try to get a grade that's good enough to go and do a PhD, and once you achieve that, you go look for someone to do a PhD with. this was the height of Thatcherism when I graduated. Britain was was in industrial decline. It was a very depressing time. So I decided to go, move north and go to Scotland, to Dundee, to do a PhD up there. And I was extremely lucky to be given a chance up there with a couple of guys. One guy's called Professor Philip Cohen, he's called Sir Philip Cohen, and another guy called Graham Hardy, let me work in their labs and get a PhD. And Dundee is an unusual place. It's a town on the northeast coast of Scotland. It's quite a spectacular place, really, to go. So I was really fortunate there. Got my degree, met my wife there, and then we decided to come to America. Right, so we come to America with one suitcase and go to Seattle, and I get to work with a really wonderful person called Edwin Krebs, who got the Nobel Prize in 1992 with his close friend Eddie Fisher for discovering how essentially a whole field of how proteins are regulated inside cells in response to all kinds of signals. It's a process called protein phosphorylation. It's led to a big explosion. They actually did the work in the early 50s and got their Nobel Prize in 1992. So for that body of work, and people describe them as the quintessential gentleman of science, and that's absolutely true. And that was another positive experience for me. We love Seattle. People are going, well, it just rains in Seattle. And I said, well, have you ever been to Scotland? I never knew rain came straight down. I just thought it came at you straight from a 40-mile-an-hour wind behind it. And it rains all the time there. But in Seattle, they had something like 220 days of sunshine. It's like, so to us, we were in sort of heaven, really. It sounds crazy, but we really loved it. And then, from there I get my postdoc. We had a fantastic time, met some fantastic people, really fell in love with the US. Its culture. You do have a culture, by the way. It's great.
Thank you.
Well, it was like a breath of fresh air coming from Britain. I mean, there are certain, like my family background is working class. My dad was he worked for the telephone company, he used to call the post office. There is a class hierarchy in the UK, and it's still there, really. And, they've done a lot to blend it out a bit, I think, than they used to. But back in those days, it was very much in place. And you felt that what your career as an independent scientist was always going to be sort of under the thumb of the establishment, and in the US, you just don't give a crap. It doesn't matter where you're from. If you can, if you have a dream and you want to make it happen, there is no place in the world that you can do this. You have no idea. I don't think people growing up in the US, Americans, really understand. You step outside this country and you will be constrained in many places by your background and where you come from. By Europe is pretty a good example of that. Japan, so US and Canada, this is also true of Canada, which is a lot much more similar to the US in many respects. It allows you, if you wanted to, be anybody you want to be, you really can be, so, and academia is a really good example of that, I think. For me, we left Seattle, became a professor at University of Virginia, and in that time, my wife integrated into. She's from Glasgow, which is, I don't know if you met anyone from Glasgow, but so my wife now runs she's chief of radiology and chief acting medical officer at the Durham VA, and everybody, she's done a wonderful job there, and she's got this very strong Glaswegian accent, and actually, she's quite tough, and my friend Neil once said to me, "Where did you meet your wife?" She's like really quite, aggressive. I said, "Oh, in a bare knuckle fight in Glasgow." and he had to
whip your butt.
Yeah. Just to put some context into who you're dealing with, so I think he believed me. I don't think I ever got a chance to tell him. I sort of made that up a little bit. But one thing in my house is you don't show any signs of weakness.
So your wife is Scottish and you're English.
It's a mixed marriage, yes.
Yes. So talk
that's
to
why
us
we
about,
have to leave. Yeah.
So Dr. Haystead, I'm interested in how you developed a passion for biochemistry when you grew up in this blue collar environment with your dad working for the phone company.
Yeah, I guess it was, we sort of lived on the edge of the countryside there, and got on my bike a lot, I like bugs and things, and I like birds. I used to love going to the ocean, especially, and I became a surfer. And just the nature, just like appreciation of it. And wanted to do that, wanted to do something in that, didn't know what. I looked at crop sciences and agricultural things for a while, which would have been kind of neat, actually. And then decided that I really like the, there's the bringing together of chemistry and how you make a drug, how do you make it work in a cell to actually do something that can help people. That seemed like an amazing, like climbing Everest or something.
I agree. But so you had that passion during your childhood, meaning did you have that passion for learning how a drug would positively or negatively impact someone's biology during your childhood, or was your childhood experience and that passion that you developed a little different?
Definitely different. I had no possibility of understanding anything as a kid, I just liked animals, I would be a vet or something, or I like growing plants, why do they grow like that? Just stuff like that, and then I love the ocean. I just like being in the water. If I see a dolphin or anything, a shark or something, I just like, wow, what's, wanted to do more, something to do with that. It could have been a zookeeper, who knows, at that time, you don't know. And then you try to, my favorite class in school was always biology or the science class. So I'd say maybe it comes from there. And, there was a teacher there that was pretty good. I remember the only one I can remember from my school that I liked was a guy called Mr. Jenkins. But the rest of the school was like being in prison. It's a hellish place. Millbrae Secondary Modern School, terrible.
So talk to us about how this passion for nature and animals and ultimately biology in your lower school education brought you to biochemistry as a college student.
Yeah, well, it all comes down to how things are put together. You can see the animals running around and the plants growing, and then you become, well, how do they do that? So in such a coordinated manner. And I guess you start to learn about cells, and well, that's pretty cool. They talk to each other and turn into different limbs, organs, and things. I think physiology was like that. How we regulate our blood pressure, and just about everything about it, was fascinating to me. And I liked doing on top of that. So you've got all that on this side, and then when we go into biochemistry, which is sort of taking the fundamental components of cells, which are the proteins and enzymes and the DNA and all that stuff. I used to love those practical classes where you kind of mix stuff together, or we had to purify a protein from a tissue extract of some kind. That's what really probably got me into biochemistry. I love that because you could kind of, you have these things called chromatography columns, and you sort of ran these cellular goos over them, and then you could look at them, you know. And I thought, well, I like that a lot. I love the practical aspects of that, you know. And that's when, you know, then you're like, so, well, I want to work in a lab now, you know.
So talk to us about that. So what ultimately triggered you and your wife to leave the UK and come to the US? Was that in some ways related to the class system that you were working in the UK, or was there something else about coming to the US that triggered the move?
Well, it was an adventure, but we also wanted to kind of escape, you know, the shackles that the oppression that we were under. No, we were really, it was an adventure to us, to be honest. We really were, you know, one of the great things about a job in science is that it allows you to travel, and you meet incredibly interesting people that for 90% of the time are loads of fun. So that, you know, international, it's very international. You get to work anywhere in the world if you want to, you know, and depending on what your interests are. There's nothing quite like it. So it's you know, job-wise, especially, you know, we were talking about the early 80s, you know, no internet, nothing. It was really quite, you know, fun. It was really an adventure, you know. Maybe the world is smaller now, you know, but it was quite, you know, you had to take several planes to get to Seattle, you know. And you were really there. You didn't come back for the weekend, you know. And
It was a commitment.
It was a super adventure, you know. So number one, and then the other one you go is you say, well, that's all very well to go have a good time, you know, but you needed to, if you wanted a career, you know, I'd done my PhD. If I wanted to become an independent scientist, where I worked had to be a wise decision, and I ended up working for Dr. Krebs, and it was a wise decision for me, probably not for him, but it was for me. You know, it really helped me, and it taught me a bit about different styles of the way to run a lab. And so that was good, in contrast to my former mentors, which are quite strict in the way that they run their operation. Let's call it that. It's very organized and hierarchical. When I walked in the door, and my first day, it said, well, Tim, what do you want to do? Like, what? I was waiting to have an answer, right? It took me about a week to give an answer, like, was it coherent? And it was like, well, so go read all about what Ed's doing, and then try to find some part of it. I like that.
So give us some insight into, so you had your week to contemplate what you were going to do now that the shackles of the British system were removed, and you were allowed to make a decision about what you were interested in. So what were you interested in, and what did you learn during this period of your career where you were studying under a prize-winning scientist?
Yeah, the first thing was, all right, I liked, while I was in Dundee, we worked with a small molecule that causes shellfish poisoning. And it, excuse me, and it is found in a little algae. It's called ochideic acid. And what it does is it attacks an enzyme in our bodies that causes muscle cramping in the guts, and it gives you, you know, the shits, essentially, to put it frankly, or and you throw up. And it's targeting an enzyme in your gut called the phosphatase. And at that time, these were enzymes that my former mentor, Dr. Cowan, was a recognized expert in and was leading the field. So I was quite interested in this little poison, and so I sort of started to work on that in Dr. Krebs's lab, but looking at it in the context of regulating the signaling pathways involved in cancer and insulin action, which nobody else was doing. And I would purify this stuff from these organisms and use it. I loved it, because you could isolate it yourself and sort of purified it, and then started to look at it in cells and what it did to signaling pathways that were controlling growth, and ended up writing a paper with Dr. Krebs on that story about how these phosphatases would control these other signaling proteins to damp their effects and things, and that sort of actually started me thinking about everything I was going to do in my subsequent career. I got interested in these, small molecules, we call them. This one was a natural product called oxalic acid, and it turned out through other people's work there were several of these in nature that were poisons that algae produce. There's I'm sure you may have heard this before, but there's, in the microbial world, there's this battle, chemical warfare going on between these communities. You find it in coral reefs to soil, everywhere. They're producing all these chemicals that attack each other's proteins, really. And unfortunately, animals and ourselves get to eat those sometimes, and the consequences are dire. And they're constantly evolving with each other. So there's sort of like this drug discovery process going on, driven by evolution in the soil and in the water, and that's a whole field. It's called natural products chemistry, which is something I wasn't going to pursue because I thought there was a better way to do this, which pharma had realized a long time before me, to make things synthetically, not naturally, because natural products, these are quite complex molecules that nature derives over how many millions of years, but I like that. I mean, I like trying to figure out how these poisons worked, and the phosphatases we were working on turned out not to be good therapeutic targets. So during my independent career, let's call it that, when I leave Dr. Krebs' lab and I go to start my own lab in Virginia, at the University of Virginia, in the Department of Pharmacology, now I'm looking, to try and start something else, something new, which is, so, to try and put a stamp on what I do rather than follow. That's the transition between the postdoc. Postdoc kind of, you get inspired to want to become independent, make a career. So the postdoc is sort of a critical time in a scientist's life, right? You have sort of, I guess, 4 paths. One is you get out of it because you hate it so much, you go do something else. You go into industry, or you try to become a researcher, and you
So Dr. Haystead, why did you choose the University of Virginia? Why was that the place you decided you were going to begin your independent career?
Interestingly, as with all things, luck, playing a game. That was a bit of a coup for me to do that. I mean, I was very, very fortunate. When you work with a guy like Dr. Krebs, you get to meet people that are influential, people in the field that are leaders in the field. Ed gave me a number of opportunities to speak publicly, as a postdoc on his behalf, which is like a huge deal. And then I managed to get a speaking slot on the work we were doing on these phosphatases at an international meeting. There's a FASEB meeting they called in Colorado. And so on the bill in front of me was the chairman of a guy called Jim Garrison, a wonderful man, who was the chair of the department I ended up working in. He gave a talk right in front of me, and he talked about some of the things we were doing with this oxalic acid. And then I talked about, the next talk was me. So there was one. So when you imagine when you submit your application, that person doesn't go, the worst thing that can happen to you is they go, read this and they go, who? So you can say, well, I met that guy, right? So, and then the other piece of luck was a guy called Tom Sturgill, who was also at the University of Virginia, who was at that meeting, and then shortly after came to Seattle, and he gave a talk, and we went to dinner, and he told me about, they were looking for folks at UVA, And it's all over that place. And another Nobel laureate, so in fact, they've had 3, you know, that came out of there. Was that guy called Al Gilman, who was famous for discovering what's called G protein coupled receptor signaling. So that was good. And, you know, Barry Marshall came from there. I don't know if you're familiar with Dr. Marshall. Yeah, he was there when I was there. You know, they didn't give him tenure. I couldn't believe that. But they regret that. Fantastic person. Absolutely, you know, we all know his story. Anyway, so it was a good place to go. Okay.
And obviously, again, one of the top schools in the U.S. You seem to have an affinity for the top schools. We'll get to that in a minute. Your time at Duke, but let's focus a little bit more on your time at UVA. So you're at the University of Virginia, and what work are you doing there, and how is that different from the work you were doing in Seattle?
It started out with well, first, Ed had said this to me. He understood if you wanted to continue to work in the same area as him, but he didn't think that was healthy to compete with him. And I agreed, you know, that, you know, you need to find your own niche, you know. So I went back to my days, undergraduate days, and I really quite enjoyed muscle and physiology type things, and try to combine that with biochemistry. And again, super fortunate, always been luck, always plays a game. And there's an old saying, and it's my motto, it's become my motto, that, you know, luck favors the prepared mind. You know, I say that over and over again. Louis Pasteur said this, probably more elegantly. But there was a couple of folks there, Andrew and Avril Somlyo, who were famous in smooth muscle biology, which is all your vascular. And I worked in I had a protein kinase background. I worked on these phosphatase things. They had these probes we were developing to manipulate those phosphatases. And so together, we all got together, and Avril, who's a wonderful person, sort of mentored me while I was developing as assistant professor, and we got some grants together, and it was really good, and it enabled me to get interested in these other proteins called protein kinases that are the signals that change muscle contraction and affect its plasticity and its physical properties, you know. So sort of, and I quite like the idea of developing tools, compounds, they're called probes, like these phosphatase inhibitors that would target these enzymes called protein kinases. And you know, and I liked it also because working with Andrew and Avril, they had this physiology that they had set up. They could take little single muscle fibers and put them in little devices that measured the muscle contractions. And the idea then was I could add either the proteins to those little fibers to see what they did, or drugs that we were making or thinking about making to see what they did. You know, so you have like this evolution of drug development now that's became, you know, what we've been doing ever since, really. So that's it was a progression of, you know, it was no eureka moment necessarily, other than a lot of good luck. And you know, that went really well for me, and I love those guys. I mean, the UVA in general was a huge positive for me, and it was like cutting an arm off to leave that place. And I felt like a big shit for doing that. You know, they help you. But the nature of this business is you have to keep pushing forward. And sometimes, you know, if you're constrained in that, you're not going to be happy.
So why don't we talk about that transition out to Duke University, the home of the Blue Devils, one of the top basketball programs at Mike Krzyzewski. Talk to us about what attracted you to Duke and caused you to leave UVA.
Ooh, yeah. So we're doing all this muscle stuff, and then an emerging field at the time was something called protein kinase inhibitors, and these were going to be drugs. So it turns out these protein kinases, there are 580 odd types of these enzymes in cells, and they control most of what we do and what we are, from everything in our head to in our bodies. Many of them are associated with diseases, and particularly cancer and autoimmune diseases, lots of maladies. And people for a long time now have been focused on them as drug targets, a new type of drug, which is called a molecularly targeted drug. And they're challenging targets, and pharma has embraced them and hated them and re-embraced them. It really, and some of them have become successful drugs, and they're out there. But I was quite interested in that. And so this is sort of like, where are we then? Mid-90s, moving on. And at this time, the biotech revolution is beginning. And it begins with the excitement around it is in the Human Genome Project. I don't know if you remember that one, in which, industrialized sequencing of human genomes begins. And out of this is why we have things like the Moderna and the Pfizer vaccine, by the way, the biotech, and our wonderful guys out of Canada that came up with the technology that allowed us to have this vaccine, which has changed. I think it has changed the face of drug discovery and how we view infectious diseases in the future. I think it's going to be a positive. One of the positives of COVID has got to be that recognition that human disease through infections is important, if not more important than cancer and other things. So there's some changes that are coming around the corner. We're in the middle of a big change. But anyway, the Human Genome Project's happening, the biotech bubble. There's all kinds of crazy stuff going on, people investing in biotech. So in my lab, we had developed this methodology to screen all these protein kinases at once, and you could catch them all on a chemical resin that we had synthesized based on a product called ATP, which is a natural thing in cells, and we linked it to a resin. And what we found with that was that we could catch all of the proteins in a cell, not just these protein kinases, but all the ones that bind to this protein this small molecule called ATP, which is the fuel of the cell. And it's about 2,000 things. We get them all in one place, and then we start to interrogate them against chemical libraries, which are small molecules. And you can create these chemical libraries from a process called combinatorial chemistry. Sorry to drop all these, but this is the basis of things of what modern pharma did at the time and still does. It screens these big libraries against their favorite targets. In our case, we catch it on this big resin, and we can screen them all at once. And to me, I recognize this as huge utility. And I've sort of outgrown my lab at UVA, and it became a conflict really with the university in some respects, because UVA is a very traditional place, and at that time they were not so amenable to the idea of doing a spin out or forming a company, because I also recognize that I can do all this academic stuff, but if we make something, if you want to get it into people, it has to get into industry's hands at some point. So we sort of hit this. And so I was giving this talks, and I'm going around, the usual, talking at meetings, meeting people. The University of UT Southwestern got interested in what we were doing, and were interested in hiring me out of UVA. Other people at Duke see that, and to cut a long story short, I end up coming to Duke. It also suited my wife, most importantly. She just graduated from a medical school, and she does radiology, and the Duke's radiology program is the top place to go, so it made sense from that point of view. But Duke also allowed me to form a company, and we formed this company called Serenex. where we house this platform technology, and we got venture funding to support that. And we even did it in a garage in downtown Durham, opposite the YMCA. It's quite a cliche. It's a real garage. It really is. I can show you a picture. It's still there. And we put a company in it.
Amazon and Haystead start at the same place in the garage.
Yeah, well, I think on a tiny scale, you can make that comparison, I think. But literally, we did. As it's a cliche. Every startup begins in a garage. I mean, but it did in this case, and it wasn't intentional. It was quite hard to find space at that time, believe it or not, in the RTP, Research Triangle Park, to put a lab in. So we put in a garage, and it lasted 8 years. I owned it all to owning hardly anything at the end, but it was fun. For most of the time, it was fun, and they made something. We actually used the process, and we made this inhibitor that targets a protein called HSP90, which was quite a hot target at the time, and still kind of is. It went, Pfizer acquired in 2008 by the company, and the whole thing evaporates right there. But we did put something. We made pills that went into people. It wasn't all bullshit. And there's that's the bit that Neil attracted Neil.
So let's pause there for a second and talk about the process that you created and the importance of you setting up your private company. So it sounds to me that what you're sharing with us is you left UVA and you came to Duke because Duke gave you the dual opportunity of continuing your academic path, but also supported you in setting up your company. So the name of your company, you said it was Serenex, and what was the spirit of what did that mean, and why did you choose that name?
Oh yeah, right. So that's interesting. Yeah. So Serene is peace of mind, and serendipity is a happy circumstance, right? So and good fortune. So that was really where that comes from, because we were trying to argue that our process allowed us to define what we call chemical starting points that were intrinsically selective for our target and had less off-target liability. So a lot of drug programs, lead molecules as they're developed, have off-target effects or side effects. Our process, we were claiming, and I still believe it to be true, would identify chemical starting points, let's call that, these small molecule starting places that were intrinsically selective for the enzyme we were interested in, and had a built-in, it removed their liabilities by being very selective at the beginning of the process. And that's what we were trying to say discriminated us from that. So that's why Serenex, it was sort of like serene being the Latin for peace of mind. Somebody's going to correct me, but I think that's true. and serendipities, a word that was coined when somebody saw Shangri-La or something, or I can't remember the name of the country. Sorry, but it's for good luck, and good fortune and all that sort of stuff.
So you created a process that would identify or limit side effects at the outset.
Yeah, essentially, yes. Yeah, it was. There's a process involved in the development of a drug that now is, it's an empirical one we use, but it uses a lot of molecular biology and structural biology, and to find something that mimics something in nature and make it synthetically, and then have it behave in a drug-like fashion and be selective is very hard. But it's a help if you can find something at the get-go that has what I call intrinsic selectivity for the target, and then its liabilities down the path then become less, and then you sleep nights in knowing that you have a pretty good within yourself that this shouldn't hurt somebody. It should help them.
help them the way it's intended. So talk to us now about your time at Duke and some of the influences that you met at Duke. You seem to have this career where you're constantly meeting leaders in various fields, regardless of the place that you go to. And when you're at Duke, you meet one of the stars in the Lyme community.
Yeah, I'm drawn to them, or the other way around. I think they come to me, and I'd like to think that. Neil was the so I got into the Lyme business, if you like, about 5 years ago with Neil. I had
and this is Neil Spector.
Yeah, Neil, Dr. Spector. Yeah, but I know Neil a long time before that because we both worked in protein kinase, mediated signaling, and drug development in that space, and I had been developing my own program around Serenex gets bought in 2008. We'd actually made a deal in that company with Neil while he was at GSK to look at one of his kinases, and he told me that we did indeed that they'd given us one of their lead molecules, and we tested in our system. We identified some off targets with it and told them what was wrong with their molecule. And he said, you were actually right. So he got to test our system, if you like. And I think that gave him a little bit of faith in me that what we were saying was real. And so we've sort of met before he comes to Duke. I'm already at Duke when he comes there. And I've been working on a new set of molecules that target this heat shock protein, HSP90, and had worked with a group of physicians that enabled us to get molecules we made against that protein, that form of that protein, into clinic. We put 2 in there that are in clinical trials in phase 1 that allow us to image. We think it's a treatment for metastatic or detection of metastatic disease and a treatment path. And Neil was familiar with this stuff too, and he's listening to this stuff. He listens to my talk. He's one of the few people in the audience that isn't sleeping. So I think he was an insomniac. So anyway, so he came, I guess. Let's see, when did he really come? Maybe 5 or 6 years ago. He said, we just got one of our new molecules into people. We were down that path. We were on a clinical path. And he just asked me, could you do this for Lyme disease? And I said, yeah, what's that? I knew what Lyme disease was because my wife had gotten it. And I got a great story there if you want to hear it.
Definitely.
All right. She's the toughest person on the planet. Okay. So one day she comes in and she's got like this red blob on her back of her calf. And she goes, oh man, what's that? I think that looks like a tick bite. I said, that looks like Lyme to me. It wasn't a bullseye, but it was a red. It looked weird. Sure enough, she gets the test positive. The only problem is she's in the first trimester of our daughter's of a pregnancy with my daughter, now 18-year-old daughter. So the problem with that is, and this is a huge issue, and I don't think people bring this up enough, you can't take doxycycline if you're pregnant, right? It's tetragenic, right? So remember that. So what do we do with pregnant women? So if you start reading the literature, which I did, there's a few papers showing increase, untreated Lyme in women that are pregnant can lead to early miscarriage, spontaneous abortion, late-term abortion. Not abortion, that's the wrong word. Miscarriage. Apologize for that. So it's serious, right? So what's the next recourse is, I think it was ampicillin they gave her, which is not a soluble drug. It has to be given by injection. And which was in the backside, and it's quite a big volume, and you've got to have that every day. And so it was so painful for her that she said, I'm going to lose this baby. This is stress. It's terribly stressful. So the next option is to come in every day for an IV infusion, sit there for a couple of hours a night or whatever. And then she goes, well, I'm a physician. I can do that myself. So she's sitting at the kitchen table every day with a stopwatch in front of her and this big syringe. And she's just slowly infusing this thing into her arm, knowing that if she pushed it in, you'd pass out and kill yourself. So I'm just thinking, we're males, we can't have an opinion even on child raising. But I'm just thinking that this person really knows her stuff. Most of us would be like, oh my God, it's going to hurt the baby, it's going to come out with multiple heads or something, but she really knew it. You just thought, holy, this person really knows what she's doing. If you're prepared to do that to yourself in that situation, I really admire that. I've never, I always remember that that's how tough she is. I think a lot of people would be freaking out over that, or would go into the hospital. But she was on rotations. She didn't want to let her team down. So I can do this myself. I'm going to take 2 hours out of the day. I'll sit there and and then it's how's the baby. And our daughter is now a student at Duke, so everything turned out great. It was just like her mum.
So Dr. Haystead, what was the antibiotic or the drug that she had to infuse into her arm every day?
I think it was ampicillin. I'd have to ask her. She's working the day, so I couldn't tell you. But I think it was. I could be wrong, but it wasn't oral. It had to be given IV, and it doesn't cross the placental barrier, so it's safe to take. In pregnancy.
So I want to touch back on Serenex and the sale of Serenex and then your work with Dr. Spector. But before we go there, I just want to highlight what Rich and you were discussing, that you literally discovered this drug discovery platform that changed the way we look at drug discovery as a society, essentially, right? So what is different about your drug discovery platform, which I believe you call this proteome mining technology?
Yeah.
And how did it streamline the discovery of drugs?
Yeah, it's, that is but one example of the way that people look at the way we go at drugs. I've used it, and we continue to use that process. I formed another company recently called IDIS Bio that's used the same process to develop an anti-inflammatory agent. So for me, it has worked out really well. And academically, we've been funded heavily since 2008 to continue the work. We made these new 2 things that are in clinical trial at Duke. We've got great big plans for another version of that for cancer and breast cancer. And a lot of all that has been morphed into a new area for us is this infectious disease play. But there are other platforms like what have the same kind of thoughts. It's not, to be fair to everybody else, people like they call it omic approaches to drug discovery. They're trying to look at lots of variables at once and finding ways of crossing them against each other, if you like, to filter out new drugs and things that could lead to new molecularly targeted therapies. It's not. It would be wrong for me to claim that we're the only people doing it. We just happen to do it this way, and it's somewhat embraced by other people in other places. There's a couple of other companies that sort of do a similar thing, out there. It's not necessarily just me. I think it would be a bit much to say that we had changed the face of drug discovery. I think most people in pharma would balk at that. There are ways that it's done and there are ways that I do it, and a way, you know, that is amenable to someone like me in an academic lab, and allows me to, you know, spin things out into companies. It's sort of a bit of a unique thing, you know. It's but there are other platforms out there. Some of them are much more industrialized than what I do. I mean, they're very complex things, but you know, they're in the same vein and the same spirit.
So I understand that you're, you know, I think you're being a little too humble. Just so for our listeners, I mean, you've been awarded grants by the National Institute of Health. You've been awarded grants by the Department of Defense, by the Stephen and Alexandra Cohen Foundation, right? You've been awarded grants by the Bay Area Lyme Association. So clearly, your drug screening platform that you've developed has seeked the attention of major players out there, and it's something that's changed the way people think about drugs in many industries, not just the tick-borne disease community. I mean, you've been working in the cancer community, you've been working in the autoimmune community, you've been working in the HIV community, right? I mean, you've taken this platform and you've brought it into many different areas of disease and have had success, correct?
Yeah, I would count success when we get a pill into people safely. And until you do that, you can't say anything. Really, you can say all of that, but until you, so if, you know, if my last thoughts as I'm passing away somewhere would be that, you know, you could say, yeah, I did that. If you can make a drug, it is not the same. If you make a drug and you get it into people, you get what's called an NDA, and it's a real drug. We are still on that path. Till you do that, you can't really, you know, have the parade and say it was a win. You know, that's why I said it's a process, right? It from my perspective, it's been wonderful. You know, I've run my lab. We've got tons of resources. We've got these big missions we're on to get to where we want to go. But it's not, the battle's not over. And, you know, I think too many of us are sort of self-congratulating ourselves on, until you get it over the line, it doesn't mean a thing.
Well, let's talk about that a little bit, though, Dr. Haystead, because you did sell Serenex, your company, after 8 years of working to Pfizer, right? I mean, this giant, and Pfizer now, I believe, has been taking your work and working towards getting it into the industry for a cancer treatment, correct?
No, not so much. I don't think that's true at all. I mean, I think that Pfizer took on our drug and sort of then got out of the HSP90 business about 2 years in. It's typical pharma. They get excited, they pay a lot of money for it, and then they lose interest in it. And actually what happened with that drug was that it was taken up again by the original VC group, who championed it all the way to phase 3, where it's now languishing at, it's called SNX 5422, and it's languishing in phase 3 looking for money to get it over the line. You know, so it has passed from my hands a long time ago. You know, that's exactly what you wanted. What we do, what we try to do is get things that have value, that work in people, and it has to be handed to somebody that can get that over the line into people. And, you know, so it has to work. You have to be able to, you're not going to give it to a big pharma organization. They're going to practice it every day. The caveat is that pharma is a business and it wants quick returns, and it's a very fickle person to, you know, hook up with, because they'll change with the wind. They come in and out of the business. And that's the negative part of it all, because your baby that you believe in, you know, the person that took it from you might have been a believer, but if the regime that they're in, the wind changes, then it's gone. You know, that's, many a drug have gone that way. So,
well, I think the good news is, though, that you mentioned the venture capitalist who worked with you in the beginning is now picking this back up and bringing it forward, it sounds like, correct?
Well, they were, yeah. They championed it from 2010 all the way up to now, and they're trying. There's a wonderful guy in charge of it right now, a guy called Steve Hall, who just believed in this for so long. Is really one of the fathers of the whole thing. You know, without him, Serenex wouldn't have got as far as it did, no doubt about it. You know, it's always a team effort, I say. That's not just somebody. The reason I work with other people is that this is what I do. And that Neil was had this perspective, farmer's perspective. Together, we were a team. We worked well together. So you got to try to bring in people. Like in my lab, I have full-on medicinal chemists. I am not a medicinal chemist, but I understand what they do. To me, they are everything. Everything in my lab is because of them. I have a wonderful guy that works with me called Philip Hughes, who just is a brilliant medicinal chemist. I'm so lucky, and I've had 2 others like that. And so it's always a team effort. So what I do I could only parade about it. If 5422 became a drug in people and was prescribed, we would declare victory. You can't declare victory if it's not there yet. It's close, but until it's there, you can't shout about it. In my opinion.
In that case, that I applaud your vision to get these drugs into people to actually make changes, real powerful changes in people's lives. But I do think that there are other things to celebrate. I mean, your drug discovery platform is something to celebrate, right? And talk to us about that, because we've read a lot about you, and we've read that you talk about the drug discovery process is very manual, and it's really all about luck. And your platform and your technology has allowed you to streamline that process to increase the odds to be in your favor. So how, in lay terms, how does that work? What is your technology, and how does this streamline the drug discovery process that you are now using in a wide variety of disciplines?
Yeah, so actually you hit it nail right on the head there. And actually my friend Steve Hall, when we interviewed him, said to me, "You make luck work for you." That's exactly true. That's exactly how it works. It basically takes 2 variables and crosses them against a common denominator, which is the ATP molecule, and it filters them. And that allows us to find these high quality lead molecules that you can really do something with. So, from there, we will employ structural biology and a whole bunch of modern tools to advance the project. And we can do that within an academic setting because of that. But the hard part was this crossing of the 2 variables. You've got all these variables on one side, all the proteins that the drug could interact with. And then you've got this thousands of collections of these molecules. And our common denominator in the middle, which is the ATP, with these proteins bound to it, you could filter it through there. And you're sort of looking at probability, right, to find something that interacts, and then measuring that interaction in terms of its affinity for the target, potency, that would be another word for that, and its selectivity against everything else. And it's that pairing. Then the 3rd variable is the medicinal chemistry part. Can you make that into a drug? And another thing that we are able to do is use medicinal chemists' knowledge. So the library on this side that we're screening of all these small molecule things, these thousands of things, there was some art in that too. There was some thought that went into what that library should be. And so we work with many talented medicinal chemists. Steve Hall was one, and there's another guy called Jim Veal that was in the company. Philip Hughes in my lab, and all got together to create this, again, to increase the probability that we would find something that would bind to things over here. So, and then the key piece was this little piece in the middle, which was this medium that you caught all this. So you really try to increase the odds and probability of finding something that you could work with. That's really, it drills down to how it works.
Dr. Haystead, you did discover a drug, this SNXS5422, which inhibited the target protein HSP90 you had mentioned earlier, correct? And
yes.
And that in itself, I think, was a major discovery, because there seems to be a lot of overlap between cancer, HIV, and possibly tick-borne illness with this HSP90, protein that you can essentially inhibit with the use of that drug. So talk to us about how you discovered that, and how can something so basic as, for people that aren't brilliant like you, How can something as basic as a protein like HSP90 be involved in so many different diseases like HIV and Lyme and tick-borne illness and cancer? And how can this one drug help all those different disciplines?
Yeah, no, it's a conundrum in some respects on the surface, because that's never going to work. But there's more to HSP90 biology than meets the eye. It functions in many diseases. It's a protein that is required for protein folding, so that when proteins are made on the ribosome, you have this big long polypeptide of amino acids. Well, this protein is thought to coordinate the way they fold up, and it turns out in cancer, tumors or oncogenes sequester that protein somehow. And if you inhibit its activity in tumor cells, it stops the growth of the tumor. The tumor cells go into what's called stable disease. Who knew? This was actually found out with a drug. It was found out with a natural product by a good friend of mine called Len Neckers up at the NCI. He was screening a natural product library. Go back to this is a thing that made by fungi, back to the microbes and things, and he found it. It's called geldanomycin, and they were actually doing a clinical trial with this drug, and I was actually involved with him showing that geldanomycin targeted this protein HSP90. And when I first contacted him back in the 90s, I asked him for some of his geldanomycin. I said I wanted to pour it over my column of ATP with all this other stuff to see if I could get HSP90, because it was annoying me. It was in the way of some other thing I was working on. And lo and behold, we run through this experiment, and we get a pure HSP90 band, pure protein coming out with a single drug out of 2,000 things. And that just turned me on to this idea works. And Len said it was important to me, and he said something fundamental. He goes, so he said something like, so HSP90 binds ATP? And I go, yeah. It turned out I'd stumbled into some controversy in the field. So together we wrote a paper about it, and it came out. And so that became my roadshow. Actually, I would use these libraries that Len had gotten for this geldanomycin, and we could see all this HSP90 stuff going on with the different types of molecules related to the original one, and then said, well, we can expand this to big libraries, and that's how we got Serenex funded. But HSP90 is conserved in every species, practically, from prokaryotes, which are bacteria like Borrelia, and all the way through yeast and parasites to humans. And but interestingly, and what's the basis of the Lyme project, and what Neil had asked me about, was if you looked at the HSP90 protein amino acid sequence, it varies between species. We all know they have their HSP90. In prokaryotes, it's called HTPG, which I can never remember it's like heat protein something G. It's a very boring name. In humans, it's called heat shock protein 90 because of its weight. But it fascinated me because if you line all these amino acid sequences of all these different species, which we have done many times, from Borrelia, E. coli, malarial forms, leishmania, compared to human, the drug binding sites where the ATP is conserved in its interactions with that molecule, but there are amino acids in between that are different, quite different. And this is a chemical biologist's or drug discoverer's dreamland in terms of now thinking about making drugs based on that target for that species, that it could be not only therapeutics, but diagnostic tools too, in a practical way that's quick. So this is what Neil was interested in and what he'd asked me about, and could we do it? And I said, yeah, we've been trying to do it with malaria with a wonderful person called Emily Derbyshire at Duke in the chemistry department here. We even published papers on it, showing that you could tease out the two species, human versus malaria in that case. And he said, well, if you can do that, can you do it for Borrelia? So, there was a swear word, yeah. And then we got into it, so, and it's the same platform. Yeah.
Sorry to interrupt, but real quick, you mentioned that you can tease out the protein for, human versus bacterial or potentially something like malaria. So is that how you use it in a diagnostic model, is where you look for that protein that's connected to a particular virus or pathogen or something like that? And that's how then you build a test around that to look for that protein that's associated with that pathogen? Ultimately,
that is what our goal is with Borrelia, is to use that target for as a diagnostic, non-invasive diagnostic, and the same target to treat. And the value, we call it companion diagnostic. It's the same idea in cancer, is that you've got this, and we've made a companion diagnostic in cancer too, and then we also have a treatment. So the idea based on the same target. So what we call the ligand part that binds to this HSP90 HTPG protein, what we're able to do with that, it can act as a drug on its own. It can inhibit that enzyme and have an effect, like it was developed in cancer. But more recently, we found we can put what we call tethers to this thing. It's like a molecular arm made of a substance called polyethylene glycol, believe it or not, same. And on that, we can attach a toxin or an imaging agent or a PET agent. And the idea then is to use that ligand with this tethered thing to develop this new approach that will allow you to, with PET, diagnose the presence of the organism in people, which I think is a critical thing for Lyme disease. To try and get at that conundrum, that we've all been trying to get at, or you guys have been trying to get at. Do I have an underlying infection? I believe I have. And if you don't, then it's something else. I'm going to ask Neil about that. We can talk about that, what that would mean. He said that's just as important as showing it's there. So, so once you've seen it, it's not enough just to see it. Okay, maybe we've done some help there. Maybe we can treat it, but the same molecule could now be used to treat it, to eliminate it. That's our big mission.
So Dr. Haystead, this is fascinating. So you're working on a definitive test to be able to tell people yes or no, you have the Lyme bacteria in your body, and you're using the same underlying technology to then work on a treatment modality to address the bacteria if you have it, essentially is what you're working on.
Exactly. Yeah.
So one of the things that I really wanted to explore with you is, we've come to understand, and I'm curious if you agree or disagree, that many people, most people in our opinion, probably all people who are sick with chronic Lyme disease, it's never just the Borrelia burgdorferi bacteria. It's never just Lyme, right? So when I was reading your research, I started to think, how is a targeted drug for Borrelia going to cure chronic Lyme? And I didn't think it would. But having this discussion, I'm starting to realize that this technology can be used to look for proteins that are associated with parasites, other viruses like reactivated Epstein-Barr, potentially yeast, which is another common problem. So you can look for all of the commonly associated ailments with Lyme and almost address them all individually and make this sort of like an a la carte approach to all of the things that can keep people sick with chronic Lyme disease. Is that a good way to think about this in the long term, in the big picture?
Yeah, like I'm writing a paper right now on this, on the work we have done that was funded by the Cohen Foundation. With Neil, we started it with Neil, and it's like unbelievable that we lost him. But something has come out of that, believe it or not. We've developed these molecules. And Neil published one paper on it before he passed away. That came out of his lab that shows, that in principle it should work. I lost my thread. Could you rephrase that again?
Sure. So I guess the first part of my question is, do you believe that people suffering from chronic Lyme disease are suffering from something beyond just an infection with Borrelia burgdorferi?
Yes, I do. Yeah, I mean, I think there's co-infections. There's some wonderful work by Michel Boombaum, sorry, from UC Davis, that shows that Borrelia infection in animal model can make those animals susceptible to flu infections. So, and these, many bacteria and viruses, and COVID is a great example, suppress or interfere with our immune system. And they do that in an insidious way. And I have no doubt that Borrelia can do that, and so a co-infection is highly likely. And this could, explain the various manifestations of chronic disease. I'm sure there's a big autoimmune element involved. And that, this is the key question that we want to ask, right? And we have to do it one step at a time, process, right? So if we can really make a probe with by PET, which is probably about the most sensitive way you could do this, see, a chronic infection in a human being, that's a big deal. It's a big deal hopefully for the Lyme community. It's a big deal because it also, in this paper we're writing, changes the paradigm for how we think about antibiotic development. For some reason I'm not allowed to use the word. People refer antimicrobial to antibiotic, but because it says that the target that we're looking at in Borrelia actually would be considered a housekeeping enzyme. It's expressed at all stages of the Borrelia life cycle, which is actually important for our approach. But inhibiting it enzymatically doesn't seem to do anything. But that's why you use these tethered things, and we can bring in these imaging agents and then the toxins or the isotopes to kill the thing. And if we can do that for Borrelia, which is quite a challenge because it's got, it's a spirochete, it's got a double cell wall. This is quite a drug challenge. If we can do that, well, why can't we do it for everything else? So that would be a game changer. So this paper we're writing right now is going to say that, and it's going to show the dramatic effects of our drug on the biology of Borrelia. It causes complete condensation of its chromosome to a single double helix right smack in the middle of the organism, and they don't come back. So a single dose does that. So then the key piece is, so that's sort of an antibiotic, if you like, or antimicrobial. If we could see the thing in the people that have the disease, then use, the drug to direct therapy, then that maybe help them. That may be a big help. And so, bringing those two things together is key, and it's all around this same protein. It's the protein is both the diagnostic and the treatment. So
I can't help but think of the work that Dr. Horowitz has done with double dapsone, and we've interviewed Dr. Horowitz, and he told us that, and it's a very, I guess, controversial way to say it, but he has a cure for Lyme disease, Lyme alone, which he believes is double dapsone. But the reason why people don't get better is because they have other things going on, maybe an underlying virus that caused, the tick-borne illness to activate, or other co-infections which aren't properly treated by the double dapsone protocol that he's using. So I wonder, with what you're developing, it may help a subset of people, but then beyond that, they may have other co-infections, underlying viruses, but the same technology can be used to target using PET technology and definitively prove, okay, we've been able to eliminate the Lyme bacteria, but you have really bad Babesia left. And you know what? We know you've done work with malaria, which is very similar to Babesia, and you target that next, right?
Yeah, I think that's the point. It's like, if we can do it for one, then people are going to say, can you do it for this one? That's the point. It's like convincing people that what you do really works. This is the difference between, being able to declare victory and just talking about it, and getting paid for it. That's what you want here. That's what Neil wanted to do, right? No messing about. The papers, they're not what we're here for. We're here to make a drug that goes and a diagnostic that's in people, and it's safe, right? Until you've done that, you've got nothing to talk about. You can, what I mean? You can't parade it around as though you're some genius, that you could only declare victory and say, what a genius I am, we got it to work.
Well, we can say,
That's
because
important.
you're certainly the genius in the Lyme community, and we're
Yeah,
grateful for you.
you it's dangerous to do that, because then you build these, I've seen it in the cancer world, these sort of rock star, strut guys that go to all the meetings, everybody, you can't do anything without their approval. And then they stifle the field for years, and they make the thinking very tunnel.
They control the narrative, and we've seen that in the Lyme community since the 70s with these, for sure.
Yeah. So, I don't speak for Neil, but I feel the same way. I think, what Dr. Hurst means well, but there are many parodies now in COVID, and it's super controversial. Chloroquine, the indomethacin, disulfiram shows up there, by the way. We see these things show up in every disease, I can tell you, in our screens. To us, they are false positives. They are non-specifics. To us, they're anathema almost. And I think that the problem with GP interactions and folks that work with Lyme, and Dr. Horst certainly does that, is it's anecdotal, right? You have to, if you want to make this something for everybody, there has to be a clinical trial. There has to be a double-blinded study. There has to be rigor in it, because what he's doing may be extremely important, but until you do that, no one's going to believe you. Pharma's not going to pick it up. You must do that. Neil was so passionate about that. We can't just give disulfiram to people and based on anecdotal studies. Let's do a rigorous clinical study. And I think some of that's going on now. I think that, I don't know how fair with all the clinical trials that are going on, but I hope that it is. Because if you can show that and it stands that test, that's progress. Yeah, but anecdotal stuff is not good.
So Dr. Haystead, you mentioned that things like disulfiram and dapsone came up in your screenings as potential false positives. So can you just explain what that means? I just want to make sure I understand that correctly.
Yeah, many libraries, there's repurposed libraries and things. It's amazing to us always how these players show up in a lot of diseases. Does that mean that they're useful for a lot of diseases, or are they just artifacts of the screen? That's the key. To us, that's what they are, artifacts of the screen. They usually have chemical liabilities. They've been around for a long time, and they're old drugs. I mean, dapsone to me goes back to the Civil War. It's like a very old drug. Is that the best we can do? To help people. I mean, if it helps some people, fantastic. But it really is off-label, and it needs to be brought, under control and with rigor. If you want the respect of the wider scientific community, that's what you got to do. And that's the problem in Lyme disease. That's where you get this, it's not real. It's
a lot more, the controversy.
Yeah. And I think that's the problem. It doesn't help that you've got the community looking around, people like me and Neil and others that think, well, man, where did you come with that idea? We see these all the time. Maybe we're throwing the baby out with the bathwater, so to speak. We should be focused on them, but to us, they're not real. There's no mechanism there. There's no understanding of how they work. They just work. That's not enough.
So Dr. Haystead, let's talk a little bit more about that, because that's a really important issue that we've identified with some of the guests that we've talked to in the past, right? So let's first talk about the relationship between the scientific method and researchers who are using the scientific method, because we're talking about that rigor now, and how we get to the point where the scientific method ultimately becomes the standard because it's accepted by the leaders in the community of people who are doing this research.
Yeah, I mean, that's it. It's a rigorous process. It's double-blinded studies. That's what we accept as real.
Meaning replication, right? So you have so Haystead does a double-blind study, and then it gets replicated by other scientists, other independent scientists, and then all of the papers that are written by all the independent scientists are now reviewed by by the people who are, again, rigorously testing and reviewing the results, and then we create a standard, right? So now when you researchers create that standard now gets sent down to the clinicians, and the clinicians are now looking at their patients and determining whether or not their patients fit within the community of people who were tested, and not outliers in the community of people who ultimately would benefit from the use of this drug. And then shouldn't the clinicians then be using other tools when they are treating a patient who is an outlier outside of the bell-shaped curve of people who are in the population that you tested?
Yeah, no, I mean, that's the, you know, I'm not a physician in any way, and they're on the ground, they're in the front line, right? So I appreciate that. And but what they're doing is empirical now, right? They're using their gut sense, and some are geniuses at that maybe, but most aren't, you know. And I think it's a dangerous path sometimes to go on. I mean, it's I mean, they're driven by compassion, no doubt about that, but you have to be careful. They need to be objective. If they get a result that looks interesting and helps somebody, that's great for that person, but don't go and extrapolate that now to everybody. Try to, you know, write books about it. You know, you're not helping. You know, you know, I think you need to take that. You do what I would do, which would, like, I'm going to explore that, and I'm going to try to understand it. You know, I'm going to try to develop it. I'm going to try to get my colleagues to accept it. And then my colleagues are only going to accept it if I show them, you know, the cohort that was done, you know, what the controls were. I know it sounds very boring and everything, but if you want to expand it, you know, make it real for everybody, that's what you have to do. It's a process. Again, I keep coming back to that word. And, you know, there are examples, you know, they're fairy tales. Barry Marshall with E. pylori is just, you know, here's a person that deserved the Nobel Prize. You know, his genius was to think that E. pylori might be causing these stomach ulcers, and he gives himself the disease to do it. He was up against this wall of people that just didn't want. It turns out there was some pharma conspiracy, and there was Zantac or something like that, one of them drugs, you know, that was going on too, but didn't help. But what a, you know, there's an example of somebody that went on to prove his point, you know, in a rigorous way, and it's accepted. But maybe people look at that and think, well, that's what I'm going to do here, you know, somehow, I don't know, but that's rare.
So, but now, haven't you given us, I mean, a community of people, a tool that wasn't available before to test the efficacy of a diagnostic and treatment tool, right?
That's what we have. That's where we're headed. Yeah, that's what we're trying to. Yeah.
So I think there's two different approaches, right? One approach is trial and error, and you're arguing that's really dangerous, right? Because even if you're having some level of success, to suggest that level of success you've had with one or two people, which may or may not have been that particular drug, but it may have been a number of other factors that we can't really control, and then try to apply that to a larger population is not the healthy way to do it. In fact, it's a harmful way of doing it. The better way of doing it, especially if you have tools like the one you've developed, is to understand the interaction between the tool, i.e. dapsone, and the bacteria that you're seeking to stop, right?
Yeah. No, I agree. I mean, if we've got this probe and it works, and we've got to establish that, that means for that to work. And I was actually, one of the things that came out of the Line Mind meeting, I was talking to Neil, he's not here now. I still talk to him, by the way. What if we made this thing? How are we going to, what's the clinical trial going to look like? Who are we going to try it on? You know, I think, you know, we really have to try it on the newly infected first, the people that we know are infected. And, is anybody doing that trial? And then John Alcott gave his talk. Yes, they're doing the trial, they're doing it. So this was like a huge relief to me because if it doesn't work there, it's not going to work in somebody with chronic disease, is it? So if we can tick that box, it's game on. Then I'm going to say, yes, we did something worthy of, a news release or something, or something to get excited about. It really works. And it's not me doing that experiment. I've given it to somebody else who's a radiologist, and they've got a patient, and they're in a trial study of X many patients, and somebody makes that correlation between our probe uptake into a zone of infection, a correlative diagnosis with something else. And in the Alcott studies, I think they're doing treatments too, right? So they're giving them things. Does it go away? Fantastic. If it's Dapsona, does it? If it doesn't, well, we learned something.
But I think we also have to identify another issue that you did talk about, which is the danger associated with creating stars in the either medical or research community. And we interviewed Dr. Alan McDonald, who's a pathologist, and Dr. McDonald had during the course of his career, he had discovered a connection between Lyme and Lewy body dementia. And he had been, and by the way, his papers, which were refused publication in the 70s and 80s, have finally seen the light of day.
Yes.
But what had happened when he initially made the discoveries and were presenting these discoveries and was presenting them at different conferences, the stars in the field at the time, Alan Steer, for example, were accusing him of doctoring data because and he couldn't get his paper published as a result of that. So I think it's really important that our listeners re-listen to the part of your very humble but important response to Matt's question about not accepting star status yourself and not having a community of researchers and doctors who are essentially gatekeepers and then force us down a particular tunnel, which is, of course, where we find ourselves now, which I want to ask you. So because we have so many doctors who are not given access and their research was not given access to academic papers, and because there wasn't a lot of money available for researchers like you until very recently, weren't our clinicians kind of limited in what they could do other than trial and error and doing the kinds of things that Dr. Horowitz and others are doing?
Yeah, I'm not going to, say that you shouldn't do it. I mean, just saying that I wouldn't do it. I mean, I hope what we do is safe. I don't want to kill anyone. I've seen people do that in our business. I have seen them do that for their career. I've seen them do it. And I don't understand. To me, it's tantamount to murder, really. I mean, you've got to understand that what we're doing first is a privilege, do science anyway. And it's a bloody struggle. It really is. I mean, you don't boo-hoo if you're having trouble getting somewhere. I mean, it could be a life's work, right? So the case you made with Dr. McDonald there, you mentioned, it is now being realized that inflammation from infections is called, in the head, is like Alzheimer's and dementia. And Neil was a big fan, I wouldn't say a fan of it, that's not the right word, but proponent of this idea that underlying there may be a bacterial infection, a viral infection. There are definitely diseases we know about, type 1 diabetes being a good example, taking out your beta cells when you were a kid and causing a lifelong disease. I mean, that's there. The realization of how these organs can use our own immune system against us is emerging in the field of immunology and in general. People are getting more and more. COVID is just this damn thing comes in and it causes this, it's the common cold virus gone on steroids. Literally, it's aggressive, causes this massive lung damage through inflammation. What did they learn early on, perhaps the treatment was not the right one. You remember people up in New York was awful, but dexamethasone, which is a potent steroid, would have helped. Helping people now. Reduce that inflammation, get them through that critical period. And then there's all this secondary infections going on with bacterial things coming in and causing long-term damage. Probably this. There's a new idea of long haul, the long haul syndrome, parodies with chronic Lyme is there. People have been talking about that. So, there's a complex, so we're learning more. So I think that Models ideas are more accepting now, but it's absolutely true that you're going to be up against the dogma. And but you've got to fight for, be honest. Don't cheat. The minute you cheat, you've lost all credibility. The only thing you have as a scientist is your integrity. That's it. It's there's this peer pressure to be the star. I just think that's the most pathetic thing. When somebody has to feel that way, especially as a scientist, go be an actor or a celebrity or something.
But Dr. Haystead, I think the problem is that one of the reasons why so many researchers and professors feel the need to, gain celebrity status is you have to find the resources to do your research, right? I mean, if you didn't have the Cohen Foundation and the NIH and the other foundations who are funding your research, and you couldn't even do your work. So there is a certain level of vanity that's required in order to be able to get the money you need to do the research you need to do.
Yes and no. I mean, I think a lot of it is, there's an art to writing these things, how you do it. I'm able to leverage what we've done in the past. When somebody said, I remember having, I was trying to raise some money, and they said that, well, what have you done? And I had the pills for Serenex. They shoved them on the table. Why is this so great? Bam, put that down on the table. We made these things, and they went into people. No one's done that. I mean, most of the academic stuff. And Neil said the same thing. That's trying to get these things that we make across the line. They must be real. They must work. We will, if they don't work, we'll walk away. We aren't going to continue to try and push that idea. We'll start something else. I mean, we could fail. I mean, we're actually going through a process the last 6 months. I said, if by Christmas we haven't fixed this, we got to start again. And it's actually to do with making a specific inhibitor for the Borrelia. There's another path I can follow, and it's to do with the biology of 90. I think we could tease the difference between the human out. But then recently it started to show promise. That we're starting to separate it, but it's a bunch of hard work. I mean, but we're not going forward unless it works. Believe me. I mean, we will not try to pull the wool over anyone's eyes over this. We can't, because the proof is in the pudding. We're going to have to give it to a guy like John Alcott. He's going to have to get a radiologist or somebody to take this agent and put it in people, that are infected with Lyme. It's got to work, I mean, and it's got to be safe. We can't get past that,
So Dr. Haystead, when you just said that you're working on something now that you just had some breakthroughs with that you were hoping to have some clarity for by Christmas, is that isolating another protein that's common with Lyme disease that you want to, is that what you're referring to?
It's the molecule backbone we were working on. We found one that looked like it was very specific. It was of the class of molecules we were working on that are known to inhibit the human form. We looked at all of them against Borrelia and found a subset that showed intrinsic selectivity and potency against Borrelia. Great starting place. Still retain some of its human, got to get those two apart. And we were doing this, what's called an SAR campaign based on a crystal structure that we got of the Borrelia protein with this drug target. It's drug rather. It was done by Matt Redembo, was also funded by Bay Area Line, and he got that thing in 3 months. He got that for us, big tick. Then we start our big medicinal chemistry campaign. We have the human form in crystal structure over here. We're trying to interrogate these two molecules. We're trying to drive away the human. At the beginning of the process, we were driving away the Borrelia one. It was like getting worse. It's going the wrong way. The human one's getting better. We learned some things about the way that the evolution has conserved things in the Borrelia and prokaryotic pockets, because they're under this constant assault from each other, and a human one isn't really. And but now we found a niche, a little chink in the armor, a little crack, and that we can get in there. And I started to find a series that looked like we might be able to separate, get to the mission, to the end goal, which was the Borrelia specific version. It turns out there's a backup plan, and that's to do with the biology of the human form, which more and more we're being convinced is only associated with bad news and disease. So if we have to go that road, but that's my consolation prize, if you like. I think if we can, the importance of deriving a Borrelia specific one is it opens the door not only for that diagnostic test, but other species. Like it will get other investigators interested, I think, in the idea of targeting first this same protein, but some other proteins. And in the Bartonella project, we're working on another protein that we think we can do the same strategy with. That would be big news. If we can change the antibiotic paradigm, if you like, that could open up a whole explosion. And it turns out that, working in this, so we actually culture line Borrelia in my lab. We got really good at it. We've figured out how to do it in a high throughput way. I've got chemists that usually work with, growing up these bugs. We've got it down to a science, so, we can really, it's become a good model now. It's sort of like a test model for other organisms, because it's, it's sort of safe to use on the pathogen scale, and become quite a good test species for other targets, I think, and expanding the idea to other things. And I think that could be something that it could, I think, ultimately even fund. You can imagine if we have this process we develop works for Lyme, who's going to fund that? Who's going to pay for that? Or maybe we could create an entity or a company that, as something you could use with COVID, or something you could use with other diseases that you could sell to somebody to create value to fund the Lyme research. Because another aspect we think to get a therapeutic and a diagnostic for Borrelia is going to be to have to drive this thing ourselves all the way to a product, and find a way to do that. Then pharma might get interested in that business.
Dr. Haystead, you mentioned antibiotics again, right? And my question is, how are antibiotics different than what you're developing? And it sounds like to me that, like something like doxycycline for acute Lyme disease, it's a very, very heavy broad spectrum antibiotic, and there's a lot of, I guess, secondary things it can do to the human body. So with your targeted therapy, I think you're saying that you're not worried about the side effects because it is so targeted, and that's the advantage of what you're looking into versus a broad spectrum antibiotic or antiviral or whatever it may be to treat other things.
Yeah, and because we use the imaging part, which is we know what we're hitting, if you like. We can see in a human, where the disseminated disease is first. So the same drug, if you like, or imaging agent is now used to bring in this toxin. One of them we call a berserker molecule. It sneaks in and it causes release of local single oxygen that really kills the organism, doesn't affect the human cells around it. So that's a little different than an antibiotic that hits an enzyme and it blocks its activity. The other problem is with conventional antibiotics is they can develop resistance, because we have this thing that targets a housekeeping enzyme. Is not under selective pressure anymore. It's non-essential to the organism, so it doesn't care whether you inhibit it or not. There may be some circumstances where it matters in its life cycle, but it isn't in its ability to reproduce, it seems. It may be a stress thing. We don't know why an organism would have a protein that it didn't care about if you inhibit it. Perhaps it's redundant. There's another way around losing that function. Probably that's the reason. So that's good, right? So we can still hit it. It's always going to be there. Then in comes something else, a toxin that in one case will generate this single oxygen species. It gets into the organism and it damages, that's the berserker thing. It causes damage of multiple proteins. That such a thing will never develop drug resistance. We can also bring in other things. We've got the imaging. We can bring in an isotope, like an alpha emitting agent, which could be used in conjunction with the PET agent, which is a cancer approach. Again, only hits that organism. Theoretically, could remove it completely, even down to the single cell. So that's the difference between that and a conventional antibiotic.
So not only is it damaging your gut and removing good gut flora, it's also allowing you to avoid having this antibiotic resistant bacteria, which we see in a lot of late stage Lyme patients. And I think that brings me to my next follow-up question is a lot of chronic Lyme patients have this disseminated Lyme or disseminated multi-systemic infection where it's in their brains, right? It's in their tissue. It's no longer just in their bloodstream. So with your approach, will this be able to penetrate the blood-brain barrier and get into the brain and get into all these spots where the Lyme bacteria and co-infections like to hide deep within your body?
Yeah. Interestingly, in a parallel cancer project, we've been working with quite a famous group at Duke that work on glioblastoma. And one of their interests is in drugs that get across the blood-brain barrier. And some of our probes we've got similar that target the human species. We're able to see uptake into the tumors in those models. And we're hoping to actually advance the human version of what will be the Lyme version, PET version, in a human study with that group. So that will give us a lot of confidence that we can cross the blood-brain barrier. We think, why do they get across the blood-brain barrier? It could be because there are transporters, and our target has, the human version has a means to be trafficked over there. I think a lot of disease, though, inflammation causes a breakdown in the human blood-brain barrier, certainly gliomas. So I'm sort of thinking that we may indeed be able to get, things going on in the brain. It'll be an interesting to see. I think the power of what we're doing, we will be able to see it. That's the thing. We will know. Let's say we can say, well, in convention, we've got to the point where we can say in conventional infections, we can see with our probes, Borrelia, we know we can see it. So let's now go look into other populations, with where we don't know why they've got dementia or they've got Lewy bodies or whatever. Do they have Borrelia in the head? So, which doesn't, which is not going to be post-mortem, it's going to be a live individual. We'll be able to ask that question. If we start seeing signals in the head that we don't see in normal people, we'll know something's going on. You either got cancer or maybe you've got an underlying infection.
So just like the work you're doing here to target the Lyme, you're confident that you can cross the blood-brain barrier because of other work you've done in the cancer world. And if not, you'll be able to see it and then work on that, I think is what you're telling us.
Yeah, there's, and again, let's say we do need to, with gliomas, I constantly talk with the glioma folks. Well, if a tumor's in the head, isn't it breaking down the blood-brain barrier? And that's why my probe gets in there. And they'll say, no, our model, it preserves the continuity of the blood-brain barrier. Okay, but in a normal human, we know our probes in the animal studies, and actually we've got a clinical paper that just came out on one of our probes. It doesn't go into the normal, head. It's not normally found in the brain. Let's put it that way. We don't see it leaking into normal neuronal tissue. So, I think that's the value of of these types of probes, and the key to answering this question that has been unanswerable before, except in post-mortem, are there active organisms in an infection in our head? Most disturbing if we find that to be true, right? I think, Neil has said, then it's a game changer, right? Everyone's going to be on this because, that's scary.
Yeah, I mean, if you can prove that things like dementia and brain-related ailments are caused by viruses or bacteria using your technology, I think that's going to open up a ton of other doors for you beyond the Lyme community.
Yeah, but when we say something like that, we can say it with certainty. We can say it it's in a testable way, right? That's the important bit. That's when you get credibility. Only if you can do that. If we have a probe that allows that to be tested, people will accept it. If you're just saying it, not going to believe you.
Well, let's talk about that, Dr. Haystead. So, I know you're still in the middle of your work and you've made a lot of great progress. And you have a ton of great organizations backing you, and you seem to be the lead, right? I mean, we were looking at some of the Bay Area Lyme, diagrams they have out there, and it seems to be you're kind of at the top as far as the researcher, right? So,
no, let's not take it. Kim Lewis is doing some good stuff. I mean, I love what he did with the antibiotic. It's a great story, actually, and it's a shame that he discovered something that was already, but it's still a great piece of work. Don't take that away. I mean, that's a great piece of work. There's what Michelle Baumbaum is doing is with immunity is extremely important. To name but a few. There are others out there that are doing things that I'm only here because of Neil. Remember that. I mean, I've got super excited about this project. It ticks a number of boxes where I think what we do could really make a difference.
But let's talk about that before I get to that, the question I was just asking. So like what Dr. Kim Lewis is doing, for example, one of the things that I think, and maybe I just don't understand the science enough, is how can a drug or this natural antibiotic, I think it's hygromycin, how can that be used in a chronic Lyme patient? Or maybe it can't be because you're just targeting the spirochete, right?
Yeah.
And we know chronic Lyme people, it's so much more than just a spirochete or Borrelia burgdorferi infection. So do you believe that discovery will actually have an impact alone to help people that are chronically ill improve their health?
I don't know, but one thing if he can in a clinical trial show it's as good as doxycycline, it's an alternative to that. That's important. Maybe it could be used in combination with doxycycline. Maybe it can be used with pregnant women, for example, because it doesn't get across the blood-brain barrier, but it's an oral drug, right? So I think you have to, we all accept the, these co-infections. That's new, right? But you can't forget where you're coming from. Let's try to get one thing done. Then we can talk about, co-infection. I mean, it almost became overwhelming to us at one point. We're well into Lyme. We're super excited about it. We've got this massive train going forward on that. I think we're going somewhere. I don't think it's an exaggeration to say I want to talk to the funders about doing the clinical trial in the next year or so. There's some things about an imaging agent that make it easier down the regulatory path. We've already done it once. The bar is a little lower than for a drug, for example. So we could really make progress there and have that companion diagnostic going forward, and then the treatment behind it. And then the Cohen's Foundation through Ben said, Neil wanted to work on Bartonella, can you do that too? And they go, well, as long as Ed's involved, Ed Bradshaw's involved, yes. And Monica, they've got all the models, so let's do it. But that is actually making progress too. But we're getting heaped on, there's always so much one person can do, and you want to do it well, right? So we're managing it quite well, I think. And meanwhile, we've got cancer things going on. And a company, so there's a lot going on with me, but but what Kim's doing is important. Don't take away from just because, oh, that's never going to work for people with chronic disease. It might help some people in chronic disease. I mean, Neil and I had this, and it's an important conversation to have with everybody. What if we make these things? We find that, well, yes, we can see, infections in people with Lyme, and in some individuals, yes, there's a correlation between you have an underlying infection. Our pro shows that it's there, but there may be some that don't. That's not what's going on. But he said that was as important as being able to say that with confidence, so that they can go and find out, stop taking the antibiotic combinations for that disease. It's not the right one. Maybe you need to look for something else on the, there's you are ill, there's no doubt about that. But it's an underlying autoimmune disease or something, or another infection, right? And that maybe they get some piece, I mean, I think it would be a while before you could definitively say that, because like I said, we need strong clinical data to back that up before you could say that. But the rigor's got to be there so that people can make an informed choice, right? That they're not living on faith all the time and the goodwill of a physician that, may have something that could help them, but I think this science needs to be introduced into this field, to make it really go away.
And it'll also help prevent all the conflict and drama and disagreements in the Lyme community. If you can use a scientific method like you're doing and use pure science, nobody can refute that in the end, right?
Yeah, I know it always be. I mean, we can't get people to get vaccinated. People just never believe anything. But yes, the main body, that's what we look for. I mean, I rely on that main body to know that if this stuff does end up in people, we're not going to hurt anyone. That would be a nightmare. We did everything we could to do it, and we got others. It's so important that others repeat what you do before you jump into that. Always have to remember that. You have to believe in what you're doing.
So, Dr. Haystead, I'm curious about your thoughts on autoimmune, because you mentioned it a couple of times. I think you mentioned one doctor who's doing some work with autoimmune, and how you could have an underlying, autoimmune problem that then allows the Lyme bacteria to get you really sick. But could it be the other way around? So I guess what I'm asking is, can Lyme trigger autoimmune, but also autoimmune trigger dormant Lyme? Does it work both ways in your opinion?
I don't know about the latter, but certainly the former. I think, cellular damage due to infection and inflammation can lead to an underlying arthritic type syndrome for sure. I mean, and I'm sure that the basis of rheumatoid arthritis in humans may have a lot of infection underlying, because it's not always, it's only a minority is it hereditary. And there are certainly autoimmune diseases that have some hereditary element in them, but very few. A lot of them, we're all impacted. The most healthy of us can develop autoimmune disease that's debilitating, and one wonders where that's coming from, right? I think it's perfectly feasible and reasonable to think it's an underlying infection that caused it. I mean, exposure to the environment, that cancer, that's for sure. We all accept that, and come back to E. pylori. What a revelation. A bacteria, no less, was causing cancer. 80% of stomach tumors disappeared overnight, literally because of that treatment. And that's the inspiration for this too, right? But look at what he did. Look how rigorous he was. Look at what he did to convince the world when the world was against him. It's a fantastic story that this was right, and he was rigorous about it. So all power to him. I mean, it's a sort of very similar story. And Neil, actually, one of his things was that inflammatory breast may be associated with Bartonella infections, and he started to work on that. And we continue to want to work on that, actually. That was one of the things we talked about, marrying our stuff that we were doing with breast cancer funded by the DoD with this, can we correlate, appearance of this surface form with a Bartonella infection in breast tissue? and transformation. That's something Neil was really wanting to work on. So again, a bit like Barry Marshall's story with E. pylori.
So I do want to ask, so it sounds like you're a pretty firm believer that an infection with Lyme and co-infections can lead to autoimmune complications. And we've interviewed over 220 podcast guests, and many of them have told us that they've developed out of the blue autoimmune diseases after developing chronic Lyme, and that as they were able to properly treat and get better from a Lyme standpoint, their autoimmune conditions actually either got better or completely went away. So do you think that that's something that is plausible or realistic, that the autoimmune condition is being caused or basically pressured by the bacterial or viral infections? And as you then treat those bacterial and viral infections, the autoimmune condition can actually reverse itself.
Yeah, I think so. And then there's some if there's a just like any allergy, right? Almost as an example that would be similar. If you're persistently being exposed to antigens, that's not good. And also you could lead to the infection might have gone away, but the damage was done. And, actually, talk about my wife again, after her Lyme infection, she developed arthritic symptoms. And she went on anti-TNF drugs, which was Enbrel and Remicade, a tremendously powerful, game-changing drugs. And for her, they worked. And she was on them for quite some time, and it was, physical game changer for her. She was back to her old self. She was exercise fanatic, doing all that. And then interestingly, and this is, I think, a new phenomenon that people are studying, is that they were so helpful for so long. It was like her T cells had forgotten these memory Ts, that underlying autoimmunity, the reason for the autoimmune disease. She does still take methotrexate and some other things, but on a much lower level than she had been doing in the past. And so one theory we have is that perhaps, that underlying post-Lyme damage, if you like, because she did respond to the antibiotic, went away with the right therapy. In her case, tremendously lucky, right? It's not, I don't know if it's completely gone. She still takes some things, but not these quite serious drugs or these biologics. So I think that, I think that if we can, in some cases, if there's an underlying infection going on, that's been difficult to detect and it's still there, and the antibiotics there, maybe they're antibiotic resistant. I don't know, they're not worked on them. We found that doxy doesn't seem, when they in our growth experiments that we're doing in the lab, they don't respond to doxycycline when they're in this, what they call the stationary phase. You can regrow them out, and they're certainly responsive in that logarithmic phase. But here they say, well, that's no wonder that it's hard to treat this disease. if you don't treat it early enough and it goes into this hunkered down state, that's going to be now it's got like a force field of biofilms around it, which are highly antigenic, probably.
Well, that leads us to a good question. It's like in your wife's scenario, it sounds like she had acute Lyme and probably eradicated the bacteria, but it sounds like it was damaged, the Lyme arthritis, and she took some of these medications like the anti-TNF that were able to help with Lyme arthritis. But on the other hand, if somebody has late stage disseminated Lyme, is it possible that Lyme arthritis is due to a persistent infection as well?
Yes. Yeah, absolutely. Yeah, I mean, it's a persistent, ongoing source of antigens or irritations to the body, and they probably affecting host proteins. Inflammation is both our friend and enemy, right? Well,
on that note, I know in 2017, at the end of 2017, there was a big in the news piece about you and some of the work you were doing with autoimmune diseases and cancer, and using this molecule, the TNF alpha, for people that were dealing with inflammation, autoimmune, and cancer. So is that where you got the idea to use that for your wife and her arthritis?
No, not at all. No, I can't say that. No, that was way before. We were just starting Serenex when this was happening. It was all, we were our own. She got it it was in our yard that she picked up the tick. And this would have been circa, where are we, 2000, Alice was born in 2003, so right then, in that first round, circa 2020, 2002. Yeah, no, way before now. No, we weren't thinking about that at all at that time. We were thinking about HSP90 and cancer. No, the anti-TNFs appeared around that time. They came out of the molecular biology revolution and everything from the Genome Project, and they were just revolutionary treatments for arthritis by controlling TNF. We have gotten interested in that signaling pathway both through the HSP90 protein. One of the things that we were quite interested in, why it worked on proliferating cells. What kind of proliferating cells do we have? We have cancer cells, we have something in the gut. T cells, immune cells, when they see an antigen, they proliferate. What do the 90 inhibitors do there? Oh, they're potent inhibitors of T cell proliferation. And then I knew quite a bit about HIV, and I was thinking, well, you know what HIV does when it infects a person? It creates a thing called a cytokine storm. It's not a very good, efficient virus. It looks for this CD4 receptor on the surface. When it infects an individual, it causes many T cells to divide with the sole purpose of increasing its probability of finding a CD4 positive T cell. So we've been working on the idea that you could use our inhibitors prophylactically to prevent early infection. Perhaps you could prevent HIV ever getting transmitted, because if it can't find a T cell within 48 hours, it's eliminated. And actually, right now there's a combination therapy called PrEP that is used. It's called the morning after pill in the gay community, and it's had a massive impact on blocking transmission of the virus. They simply prevent the propagation. We actually went with this idea of using our HSP90 inhibitor, which is safe in humans because it's given to cancer patients at very high doses. Could you use this as an anti-HIV drug? We're still trying to push that agenda, actually, and by simply, not as an antiviral, but something that blocked T cell proliferation acutely. And, actually I ended up talking to Bill Gates himself about that at one point, which was really amazing. He didn't buy it, but I still believe it to be true, that it could work, and then we've also added, that got us very interested in TNF signaling and proliferation in immune cells, and that's been the basis of our new company, which is called IDIS Bio, which looks at TNF signaling inside cells, and we believe this might be an alternative to Enbrel and Remicade. We're targeting an enzyme called TAC1, which is a protein kinase in there, and to develop a series of exquisitely selective molecules that hit that target. And we hope it'll be an oral drug that you can use as an alternative to Enbrel and Remicade, which are biologics. So, yeah, this whole platform that I work on allows me to touch on all these different fields, and we're always looking for opportunities where we might be able to use it or give it to somebody that might make a use of it. So
Dr. Haystead, you're giving us a ton of hope and confidence in the future for people that are just debilitated and chronically ill. And hopefully everybody listening is feeling a little bit empowered and having some sense of relief for the future of their health. But give us an idea of time frame and next steps for you, because it sounds like you said earlier that you're looking to find some funders for clinical trials in the next year or so. So what does that mean time-wise for better testing and better treatment for Lyme with all the work you're doing and all the people you're collaborating with?
Yeah, well, we're on a fast track right now because we have a lead molecule that it does target the human form, but it exploits the biology between the human and the bacteria, in that the human targeting part only occurs in metastatic cancer. So, that would have a dual use. Then maybe you could fund its use in Lyme. That's one business idea we've been bandying about. But that one's a pet agent, and some fortuitous events happened, as they always do. There was a paper that came out in Nature Chemistry, right, prestigious journal in April, and it described a new isotope called Cerium-134, and it's a positron-emitting lanthanide metal that you can chelate. And why is that important? So chelates are used medically already. There's a chelate called DOTA that you use in MRI for a compound, a metal called gadolinium, which is also a lanthanide metal related to this cerium. So we were quite excited. Well, you can give people this chelate and it doesn't hurt them. Good. Okay, so let's link it to our HSP19 inhibitor and our Borrelia targeting drug. And now with this new positron emitting, that's a PET enabled metal. So it's a radioactive metal that's produced right now by the Department of Energy. So it's kind of cool. You're talking to people that have these linear accelerators and working with an accelerator in Oak Ridge. And they're sending us, we became a test site for this Cerium-134. Why are we excited about it? Because it's something that you could give to a radiologist, you know, and they could take the molecule that we make, mix it with this radionuclide. So the chelating part that grabs this metal will then carry it to the tick, to the Borrelia, and allow us to image it in a very practical. It's a very high energy. It's a bit like the analogy would be FDG PET that you commonly use except this one's got a 3-day half-life instead of a few hours. So it's a high energy emitter. So it would be a very, very sensitive, non-invasive molecule or probe to detect infection at an extraordinarily sensitive level. This is like a dream for us, because we had another method we were using which involved an iodine isotope, a little bit more difficult. This one's made that path very simple now. It means that we go through the regulatory process to get into humans. What we worry about now is the chemical entity. You know, so we take all of our synthesis, that has to be under GMP, our safety testing studies that the FDA require. It's all about the chemical, not about the radioisotope. When we've shown that the chemical is safe at the doses we plan to use in humans, which is going to be a microdose, non-pharmacological. It's just a matter of mixing it with the isotope, and which is a positron emitter at microdoses. So that's not non-lethal, non-dangerous to the person. It's just like doing a regular PET. So this is now a fast track. I believe that if we can demonstrate with our current molecule proof of concept in an animal model of infection, we are now on a regulatory path with that molecule. We will go all out to move it into humans. And that can take a year to a year and a half to do with the money, which isn't a staggering amount of money you might want to know. It's probably about $1 million to $2 million to do that, to get to that IND through the safety. There's a regulatory process for the nuclear part, but that involves just, making sure the isotope itself is clean. It's not got any contaminants in it. That's handled by Department of Energy and physicists and things. So I think it would be something really worth talking about. That we would be in a clinical, and if that allows us to plug into hopefully Dr. Alcott's or somebody's newly infected trial, where we can show in a small cohort, I think you'd only need 20 patients, where you've got, let's do the basic thing. Not all Lyme infections are signified by the bullseye, but we do accept that if you got a bullseye, probably a good chance you've got so we could even say, let's do it in the mouth, where we've no doubt this person's got. Do we get a signal? Can we follow those folks? Yes, tick that box. Now you've got something that you can say, I'm going to look in people that, you can do that in a Dr. Horowitz way, which might be anecdotal. It might be in those, patients that Dr. Walcott and others deal with, let's have a look at you. This is not going to hurt you. I know, I have faith that it should find the spirochete if it's there, or if you've got, it will even, we know our probes will bind to that biofilm, by the way, so that could be another signal we might pick up. So that's my hope, and then perhaps we've got the therapy behind it.
Well, Dr. Haystead, thank you for joining us and giving us such hope for the future. I know I'm walking away from this interview feeling just energized and hopeful for what's to come in the next year or so with all your research. And please stay in touch and keep us posted so we can share that with everybody who listens and follows us on social media. And again, thank you so much for taking time out of your busy schedule to join the Tick Bootcamp podcast. Yeah,
it was fun. Nice to talk to you. Yeah, nice to meet you. And keep up the good work.
Thank you for listening to the Tick Bootcamp interview with our guest, Dr. Timothy Haystead. To our listeners, we have a call to action. First, if you'd like to learn more about Dr. Haystead and his work, please visit medschool.duke.edu. Second, if you enjoyed this episode of the Tick Bootcamp podcast, please share with your friends by using the social media buttons you see at the bottom of our post. Third, Tick Bootcamp has created a Tick Bite blueprint that has been inspired by the information that has been provided to us by past podcast guests. We urge you to visit our website at www.tickbootcamp.com to view the blueprint. Please note we would appreciate any input or any improvements you would like to share with us. Fourth, don't forget to subscribe to this podcast on Apple Podcasts, Google Podcasts, or Spotify to get your automatic episode updates of our Tick Bootcamp podcast. And finally, we thank your community for your comments on our past podcast episodes. Please take a minute to leave us an honest review on Apple Podcasts, on Instagram, or on our website. We make it a point to read every single one of the reviews we get. Thank you, as always, for listening.